All right, let's start getting started. Can somebody confirm if I have audio? Thank you, Nina. All right, so hello everyone. My name is David Ruck. I'm here at the University of Maryland in Baltimore, Maryland, and welcome to Cytometry NR. So, this week is now the 8th week of the Cytometry NR course, and it's the day that many of you have been patiently waiting for. Namely, today is the day that we're going to learn how to create gates in R for our flow cytometry data. So today we're going to be showcasing two separate approaches. One, a manual gating style, which is mediated through the flowGate R package, which is available from Bioconductor. And the flowGate package allows us to do manual gates by launching an interactive R Shiny app, which provides us an interface that allows us to draw gates, very similar to what we see for commercial software. Separately, we're going to look at how to use the openCyto R package, which is also from Bioconductor, to automate the gate setup via a CSV template. We'll also see how to modify and apply gate constraints, allowing us to visualize our data, just to make sure that all these gates are being applied correctly, so that not only are we confident with the gates that we're implementing, but others who are looking at our data afterwards can also be confident. So, a bit of walkthrough just like we do every week. If you have not yet done so, please go ahead and sync your fork of the Cytometry in R course folder, since we have updated the data sets, and go ahead and pull those locally within Positron until they're on your local computer. And then if you're keeping the course organization structure, you now have a week 8 project folder repository. Go ahead and copy over the data from the synced version of your Cytometry in R folder over to your local project folder, just so that there's no conflicts when you're trying to bring in the data again next week. And for those who have completed the community feedback survey, I thank everyone for their contributions. The one of the recommendations was describe how to do this in a video step, and that's on my plans to record for early next week. So let's go ahead and first get started by making sure we have the packages installed that we're going to need throughout today. So one of the things we can do is we can use the package version function, and basically go ahead and check each of the packages we're going to be using individually just to make sure what the current setup is. This is necessary because of ggplot2 going to version 4, changing a lot of things internally. That has caused downstream impacts on both ggCyto as well as flowGate, so we need to make sure all the correct versions are installed if we want to be able to draw our own gates. So currently, you just need to have ggplot version 4 installed. ggCyto, the bug fixes were implemented in 1.39, so any version greater than 1.39 should work. openCyto, the current version I have installed on my computer, is 2.22. And then the flowGate version, which we're going to be working on making sure everybody has the correct version in just a couple minutes, should be 0.99.2. So, unfortunately, flowGate is one of the bioconductor packages that also got caught up when ggplot2 rolled to version 4, which caused the ggCyto break, which we saw a couple weeks ago. Now that the ggCyto side of the code has been fixed, flowGate still needs to update some of those internals on its end. So if we try to install flowGate at this point in time, it's not actually operational when we're installing it from Bioconductor. So at the time of this class in April of 2026, I have not yet heard back on the bug report, which I submitted to the flowGate maintainers. So functionally what happens is if we try to install flowGate via Bioconductor, which is the way we would normally do so, it would install without any issues, but once we try to launch its Shiny app, it would immediately crash and we wouldn't be able to implement our gates. Fortunately, since this software is open source, I was able to take a look at the source code, basically figure out where the bug due to the ggplot2 and ggCyto changes occurred, and then because I had forked my own version of the flowGate R package, I was able to implement those changes, allowing us to actually be able to do the previous functionality we had for flowGate when it was the previous version of ggplot. So while we wait for the flowGate maintainers to go ahead and upstream, meaning bring in those bug fixes to the main Bioconductor project package, we can still use the forked version and have access to these changes. So how do we go about installing my forked version of the flowGate package that contains the bug fixes for what we need to do today? So if you have already installed flowGate, which is possible if you completed the week one and installed all the packages, you'll need to actually remove that bioconductor version of flowGate. And the way we do that is with remove.packages function and just providing the name of flowGate. You'll see it kind of looks like this in terms of our console. And anytime you're uninstalling a package, just go ahead. It's good practice to go ahead and close Positron and then reopen it just to make sure it's actually truly unattached from your local environment. Once we have uninstalled flowGate, we need to go ahead and install the forked version from my GitHub. And the way we do this for any GitHub packages is using the remotes package, specifically the install_GitHub function, which is what we saw during week one. Alternatively, if you had completed the week one take-home problems, you would also have the pack_r package already installed on your computer, so you could also use package_install function to be able to do that same functionality. So what this looks like in terms of your workflow, if you're following remotes, you just need to, once again, attach that package to your local environment via the library call. And then at that point, you're using the install_GitHub function, and we're looking specifically for my repository where that forked version's at for this flowGate package, and we're going to go ahead and install that one to our local computer. And once you have installed the forked version, you should see when you run the package version function for the flowGate package, it's showing up as 0.99.2, which is the one that has the bug fixes for the ggplot, ggcyto breaking bugs, so that everything is now operational. All right. Now there's going to be several bioconductor-based packages we're going to be using quite extensively throughout today, so it just makes sense to go ahead and attach all of them to our local environment so that when we're looking for functions that are present within these packages, we're able to find them without much issue. So go ahead and library flow workspace, ggplot2, ggcyto, flowGate, and openCyto. And just like we've been doing throughout the last couple of weeks, we need to go ahead and identify our data set. We'll be continuing working with our spectral flow cytometry FCS files that we started working with back in week 5. If you're keeping the course organization scheme, they should be within your data folder, within your working repository. And sure enough, this is just copying and pasting in the code that we already have. And you can see we're using the file path function to designate look within the data location. And then we're passing that storage location to list.files function, which then searches inside, finds anything that matches the pattern equals .fcs, and then returns the full .names equals true argument so that we have the full file path between our working directory and our FCS files of interest. So you can see these are the 6 FCS files we've been working with for the last couple of weeks. And now that we have our FCS files, we need to go ahead and load them into a gating set. So just repeating the code that we've seen over the last couple of sessions, we first load it into a cytoset using the load cytoset from FCS function. Once it's been created into a cytoset, we just pass it to the gating set function to go ahead and load it into a gating set object. And just like we saw last time, we need to apply transformations just to make sure we can get good resolution of our positive and our negative populations, which will be especially important today when we're actually going and applying our particular gates of interest. So this is just copying over the code that we had worked and optimized on the week 7 material. Basically, we are grabbing the floor names from the gating set. We are removing anything that has forward scatter, side scatter, time, because those are on a linear scale and we don't need to transform them. We are then providing our particular arguments for the FlowGeo style by exponential transformation, which is becoming our little transform. This transform, we're combining it with the list of individual floors to make a transformer list. And then we're applying this transformer list to our gating set object, which causes all the transformations to actually be applied, changing how we visualize our samples. So if this all went as intended, if you go ahead and use ggcyto and plot the first specimen that's present in our gating set, looking at all cells, because we're using subset is equal to root, looking at our 2-axis BV805, which is CD4, BV650, which is CD8, we can see we can visualize the different T-cell subsets, and this looks pretty good as far as resolution between our positive and our negative populations, so this is kind of like what we are trying to get out of our transformation. Obviously, if you're working with your own data set and it doesn't look quite as well resolved, you may want to go ahead and rerun everything above and then change your transformation parameters, kind of like we saw during week 7, until you get to a point where you're happy and you're just like, okay, we can move on to the gating portion. All right. The first package we're going to look at when it comes to applying gates in R is the flowGate package. So flowGate is originally a bioconductor package that launches an interactive R Shiny app. This allows us to draw and apply various tiles of manual gates, which get added into our existing gating set object. It is one of the newer bioconductor packages, and it addresses a major need for many of the R-based cytometry workflows, which makes it especially unfortunate that it got caught up within the ggplot2 version 4 bugginess. So while we wait for the maintainers to go ahead and upstream the bug fixes, we're using this fork version, but my goal is to definitely get the bug fixes to the bioconductor version, not to make a hard fork and have to maintain this myself for the long run. So let's go ahead and replicate what we've been doing for my regular workflow. So I'm most of the time gating on T cells, so I'm focused on gating down to my T cell subsets, and then looking at CD4, CD8, and double negative, i.e. CD4 negative, CD8 negative T cells. So this is what I would normally be doing in commercial software. So if I'm trying to be able to replicate what I do there in R, I kind of want to be able to go through those exact same steps, just so I know that I can actually accomplish what I'm trying to do. So within flowGate, the function we're going to be using quite extensively today is the GSGate interactive function. And this will look something like this in terms of the syntax. So what we're providing to this particular function is our already assembled gating set. In this case, I called my gating set FFC underscore gating set. So that's what I'm providing to that value. If you're creating a new gate, that's being provided to the argument filter ID. So in this case, I'm always going ahead and start off with my singlet gate. So I'm providing a new gate name, singlets. Sample denotes which sample from your gating set you want to use for visualization of the plot. I will keep it at the default for sample equals 1 until later on when I actually need to see some cytokines, which I'll switch it to one of the SEB stimulated specimens within my gating set. And kind of like what we saw for ggCyto, we need to provide an X and a Y axis. This is where the dims argument comes into play. You can see since I'm gating for my singlets, I'm using forward scatter area by forward scatter height. And then because there's no existing gates currently placed on this gating set that we created from scratch, the default will be the root gate is where we're going to assign this new singlet gate to get created and placed below it. And this was the description of what I just kind of went through. The main things are once you're ready to go, you go ahead and run this line of code. And what's going to happen is it's going to launch what's known as an R Shiny app. And as you hit that line of code, a couple things are going to happen. And the first place I want to draw your attention to is to your console window at the very bottom center of the screen. You'll see as soon as you launch any Shiny app, it will start by reading out a message saying loading required package, which is the Shiny package, which makes all this possible. And then you'll see a printout that says basically listening on HTTP, and then it lists an IP address, meaning it's listening to what's happening on a web browser. At the same time, you'll see this little stop sign appear in red, which oftentimes you'll see appear if R is running a very intense script that's doing a lot of calculations. In this case, once it launches the Shiny app, until you close the Shiny app or until you hit this red stop sign, you won't actually be able to type anything within the console window until one of those two criteria is met. At the same time, the Shiny app is going to launch. And what you're going to see is something that pops up that looks roughly like this. It's going to be within the Viewer tab on the right secondary sidebar within Positron, and also similarly for RStudio. Now, one of the things that we will see as it first pops up, it may not be in this correct dimension. So one of the things I always try to do when I'm launching flowGate is just go over here to the left side of this viewer and click and drag the border until I can see both the little widget as well as the plot in the same view, just to make my life a little bit easier so I don't have to scroll up and down and I don't have to scroll side to side when I'm trying to view the actual gate that I'm trying to implement. So flowGate allows us to draw many of the similar types of gates that we would be able to do with commercial software. So the one that I use most frequently is the polygon shape. So when it comes to drawing a singlet gate, let's go ahead and try to draw it using the polygon option. So if you're looking at flowGate, you can see within this little widget bar, there's a gate type, which allows us to draw rectangles, polygons, spans, and quadrants. We'll go ahead and select the polygon. I don't really mess with these other defaults as long as I'm able to properly visualize my current cells within the current plot. But once I've selected the polygon, what I can do is I can start clicking in one location and then go down to the next location and click again. And what's going to happen as we're visualizing the Shiny app is it's going to draw the first line. And then when I come over here and click on the next location, I can go up to the next location, keep on clicking. It's going to basically continue extending the line to each subsequent point we click on. Now, if I mess up and it's not actually showing what I wanted to show, all I need to do is come back over here to reset, click on reset, and that will get rid of all the lines, and you'll be able to start again. Ultimately, you'll end up with a plot that looks kind of like this. You don't have to worry about clicking back to the original point. As soon as you click on the Done button, those two, the first and the last points, will end up connecting together. So, in terms of saving your gate, all you need to do is, once you're happy with your gate, go ahead and click Done. And at that point, the Shiny app is going to become inactive. It's going to basically close. And what you're going to see is you go back to your console window, that little red stop sign is no longer going to be present, but you'll see some outputs get printed to the console. You'll see a done exclamation point appear, and then you'll see some information about the gate. In this case, a polygon gate singlets with 6 vertices, dimensions /scatter A, /scatter H, and a couple other informations in a list style format. So this information is being passed to your gating set, providing the information as far as the coordinates of where to place the particular gate on which particular dimensions. So rather than you having to specify, I want to place my first point here, 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 it's doing all that monotonous kind of work for you and just passing it directly to the gating set. Now, let's go ahead and make sure that it did actually correctly save, because occasionally a bug can happen. So the best way to double check is the plot function, which we've seen used to visualize our plots within our gating sets over the last couple weeks. So if we check it right now, we can see it's no longer just the default root option, but we can now see that below the root, we now have our singlets gates. And just because we have a visual gate doesn't mean it actually gated for each and every specimen. We don't know if it just gated for a single specimen, so we should go ahead and verify that. So how do we go about checking it? Well, we can use one of those functions we encountered over the last couple sessions, the gspop_getCountFast function, which we're just providing our gating set as between the parentheses, and it's going to return the counts for that particular gate. So you can see this first one is a singlets gate. We can see it had 978 cells, 9788 cells out of the 10,000 that it started with, and vice versa on the way down. So you can see when we drew that gate using flowGate, it just applied them to all the specimens that were present in our gating set. It didn't tailor it to each individual specimen. It was just a broad across the board application of a gate. Now, this is just the very first step as far as the gate creation. So if I was following my regular workflow that I was doing in commercial software, I'd want to throw in a viability gate or a scatter gate of some form just to make sure I'm getting only my cells of interest or only my viable live cells of interest. So in this case, for this example, we're continuing by drawing a viability gate. So if we're drawing a new gate, we're once again using the GSGate interactive function. And there's a couple things that have changed. First off, we're drawing a new gate, so we need to provide a new filter ID name. In this case, we're calling this gate live. Since I'm gating based on the viability die, I need to switch my dimensions. In this case, I'm using /scatter area by zombie_nir, just so that we're actually correctly looking at what we want to look at. And I want this gate not placed under root, but under the newly created singlets gate. So I go ahead and change subsets argument to singlets, just to specify where I want this new gate to be created. And then once this is all set up, we just go ahead and rerun this line of code. The Shiny app will relaunch, and we'll get this new visualization that we're then able to draw a gate on. Now, in this case, let's go ahead and test out a couple of the other gating types that we can do. So let's go ahead and use that rectangle gate, and let's kind of see what we actually accomplish by using this option. So let's go ahead and select rectangle, and this will allow us to draw a rectangle gate. And now if we look at the plot right now, you can see there's not many dead cells currently. And the reason for this is, for the data sets I provided to the course, I had done a lot of the cleaning, the pre-processing cleaning ahead of time before I saved the FCS files, just because I wanted to make sure we had viable cells, not just 10,000 debris on the side of the scatter. So there's not very much dead cells currently here. So in terms of actually visualizing this plot, you can see it's doing very similar to what Cyto did, where it zoomed in on all the dots that are present, not based off of the coordinates that you provided like Cartesian to be able to zoom out and view the whole picture. So this is just one caveat when you're viewing these plots. If you've subsetted down and you're just looking at your CD8 T cells, it'll look kind of like a uniform blob for CD8. You won't be able to see the white space between where you would expect to see your CD4. So yeah, just a general caution, the GG-Cyto plots will zoom in on the dots that it has available. But back to how to draw the rectangle. For drawing a rectangle, you just select the option, you click, and then you kind of click and drag to be able to highlight a particular area of the plot that you want to draw the gate. So what this will look like is kind of like this. You'll be able to get this formatted as you like. If you mess up and want to start again, you just have to hit that reset button. But once you're happy with the rectangle that you have drawn, all you need to do is go ahead and click the Done gate. And once again, you'll get some returns that get returned to your console window, specifying what kind of gate it is, providing the dimensions and the locations on those dimensions that each little vertex of the gate needs to be created at, etc., etc. And let's just go ahead and double check. Once again, by plotting, we can see we have our live gate now created. And double check again to make sure it did actually apply it to all the specimens. In this case, I'm using a pipe to filter for just the live samples, just so we can see those numbers. And you can see, yep, these live gates were applied underneath singlets for each of my 6 specimens in my gating set. All right. So that's 2 of the 4 flowGate options in terms of drawing gates. The other option is the span option. So in this case, as I had pre-processed the cells, everything should be T cells. So we can go ahead and do a span gate on my T cells. In this case, looking at like CD3 or CD45, just grab the entire population. So once again, if we're going down one additional level, we need to provide a new name of this gate. In this case, I'm going to go ahead and call it T cells. I'm changing my dimensions. Spark Blue 550A was my CD3, Spark Blue 574 was my CD45, and I want this placed underneath the live gate, so I'm changing out subset to live at this point. And at that point, once the Shiny app relaunches, I'm going to go ahead and select span. And when you're drawing a span, you're just basically going from one angle to the other. So if I want to create a span based on CD3, I'm going to click and drag from here all the way over to create a gate based on the CD3. Vice versa, if I wanted the y-axis dimension, I would just kind of click and drag it along the y-axis just to be able to denote what we're doing. So when I'm actually drawing a span gate, it's going to look something like this, where it's going to be a little blue rectangle that's drawn out. But what R is actually going to retrieve is basically going to be this coordinate versus this coordinate, and it's going to graph the span of data that's present between these 2 locations. And once you're happy with your gate, if you don't need to reset, go ahead and select Done. And just like we would expect, there's a little readout to the console window giving the gate information. And if we check the plot, we can see we now have T cells underneath live. And if we check to see did they actually gate and quantify the cells that are present within our gating set, we can see, yeah, we're getting fewer cells than what we started with. All right, now that we're finally down to T cells, this is where most of the time my gating scheme will branch out, where I'll start looking at CD4 T cells, CD8 T cells, and CD4 negative, CD8 negative, double negative T cells. So let's go ahead and just start drawing a couple polygon gates just to show a little bit different functionality. So in this case, I'm going to go ahead and target my CD4 T cells. So I'm going to go ahead and provide that to the filter ID. I'm going to place this underneath the T cells gate, so I'm changing this to subset. And I'm going to go ahead and switch the axes to the CD8 and CD4, so the BV650, BUV805 syntax for this particular panel. And you can see this is our visualization. I'll just go ahead and draw a polygon gate around my CD4 T cells, and then go ahead and save it just to make sure everything is saved to my gating set. And typical readout. All right, at this point, we should also just mention that flowGate uses GG-Cyto, which uses ggplot in the background. So kind of like anything that we can do with GG-Cyto, oftentimes will work for flowGate. So we saw last time with GG-Cyto that we could, instead of using the fluorophore name, we could provide the marker name, specifically CD4, CD8, as part of the dimensions, and that would actually serve to plot the data. And this also works in the case of flowGate. So if you don't want to remember which particular fluorophore in your panel a particular marker is in, and if you remember the marker name, you can provide the marker names. So repeating that same line of code, just using CD8 and CD4, we can actually get that plotted. And vice versa, let's go ahead and add an additional gate. In this case, we're looking at our CD8 gate. Now, CD8 is going to be under filter ID. We're still placing it under T cells. But flowGate also allows us to visualize an already existing gate so that we're able to see how it compares to what we have previously gated. So we have already created the CD4 positive gate, so we can specify that to the overlay gate function, which will then showcase it within the same plot. So when we render it and we open up the Shiny, we can see the CD4 gate that we previously drew is already present. All we need to do for drawing the CD8 gate is select this little polygon option, draw our gate around our CD8 T cells, and then once that's done, we can select done and proceed on. And just once again, there's always going to be that little console readout, which is quite useful to troubleshoot, making sure you actually did gate properly and set up a gate. And last but not least, we can repeat for the double negative T cells. And as you can see, this overlay gates can be provided a C, which stands for concatenate, which basically creates this little vector saying, show me the CD4 positive gate, show me the CD8 positive gate. In which case, when we're drawing our double negative T cells, we're just switching out filter ID for the name of the new gate, and we're keeping it there for T cells as the subset the gate is being placed under. And at that point, just draw a new gate and go ahead and save. Once again, get that readout, and once again, if we double check, we can see we have gone several layers down in terms of gating. So we've made some progress working entirely within R. And then if we quickly check to see the statistics for that last gate we created, the double negative T cells and filtering through the pipe, we can see these are our counts for our double negative T cells across each of the populations that we're interested in. Finally, the last type of gate that we can create using flowGate is a quadrant gate. So this can be quite useful if you're looking at cytokines, for example. So in the context of this particular panel, I have TNF-alpha, I have interferon gamma. All the samples that we provided were cord blood samples that were stimulated with SCB. So we're not going to get very much cytokine. It's going to be predominantly TNF-alpha from the CD4s, a little bit of CD8 producing interferon gamma. So main thing when we're drawing these gates is I prefer to go ahead and be able to visualize what my cytokine expression is. So we will need to switch specimens from a control specimen, which is what we're currently at, to a SCB specimen, which is in our gating set, would be the 6th specimen. So if you're checking your metadata, remember from last time, it's basically the pdata function, just to see who's first, second, third, all the way down for your gating set IDs. And then once we're doing this new quadrant gate, we're providing the filter ID for the name. So it's going to be cytokines. This is going to be underneath the CD4 gate. I want to visualize TNF-alpha and interferon gamma on each axis. And then when I want to draw a quadrant gate, all I need to do is select quadrant, identify the spot I want to place the gate, and click once. And once this is done, it's going to go ahead and apply. And you'll see this gate now is visualized. And as you're checking, if this gate is not where you want it properly, you just have to hit reset, and then you can select done and continue trying again. And I'll get to your question in just a minute. That's a great one, and we'll address it a little bit. And sure enough, at this point we get the little console readout, and gate is created. And if we check the plot, we can see that the quadrants have now been assigned. TNF-alpha positive, the 4 different quadrants for TNF-alpha interferon gamma combinations. Now, what happens if we accidentally end up misdrawing a gate and we need to correct it? So obviously, if you're actively drawing the gate and you mess up, you can just go ahead and click reset. This plot is going to lose all the little gate sets that we've drawn, and you're going to be able to redraw the gate and then select done. But what happens if you've already clicked done and it's already been incorporated into your gating set? So what you can do at that point is, if you're checking this one here, let's say I'm going to intentionally misset a new gate that I'm creating. I'm working underneath CD8 T cells. I'm creating a CD161 positive subset. And as you can notice here, I can also only use one dimension, which will return a histogram. Not that I'm recommending gating based on a histogram alone, but you can see we do get this back. But let's say I completely mangled this gate. I use the span, and now it's grabbing some of my negative cells, and I accidentally clicked done, and it's now present within my gating set. So if you want to just go ahead and redraw this gate entirely, what you're basically going to do is provide a new argument, which is the regate is equal to true. This will basically go into your gating set, delete the existing gate by that same name, allowing you to redraw another one on the Shiny app. So as you can see, same line of code that we had before, just setting regate is equal to true. And then on relaunch, we can then regate correctly and then save it so that way we're able to update. All right. So far we've placed all this effort, we've drawn out our gates, but what should happen if we accidentally end up closing positron? So at that point, we'd basically have lost all our effort and we would need to redraw our gates again, which doesn't sound like a particularly good use of our time and also does not sound replicable, reproducible, which is the main goal for a lot of this course. So we definitely need a mechanism by which we can save all our hard work, all our gating sets, all the transformations, compensations, and gates error accordingly. So if we don't want all this to go away and not be able to access it again, what we're going to need to do is use the Flow Workspace package, specifically the save_gs function, which allows us to save our existing gating set objects so that we can access them and load them into R later on. So the way save_gs works is basically you're specifying both a file path and the name of the particular gating set. So in this case, you're going to create a new gating set folder. So in this case, I'm going to call it flowGate examples.gs, just so we know it's a gating set folder. Don't mess with anything that's present within. And then I'm passing this combination file path and name to save_gs alongside the gating set in question I want to save, and this will go and save the object. And what this will look like if you're looking at it and saving it within positron is underneath this .gs folder we just created, there'll be a bunch of H5PB style files. Don't mess with these, but this is basically the representation of your gating set stored within that folder. And these can then be loaded in and brought back. Now, to load these back in is rather simple. We just need to provide the file path back to this folder that we have created. In this case, we use the load_gs function. And you provide the file path back to that stored folder. You use the load_gs function from Flow Workspace. And if you plot that gating set we have brought back into R, you can see all your gates are still present. And if you run the little stats functions, you can see that, yeah, it's still properly gated on the populations that we're interested in. So, in the examples above, when we were creating all these different gates, we were starting one gate, then the next gate, then the next gate. We were running them on individual code chunks, opening and closing the Shiny Gap multiple times, which could add a little bit of time. So flowGate tries to speed things up a little bit by allowing you to specify which gates get drawn sequentially after each other. And this is going to look a little bit different from what we've currently done. It uses the Tibble package, which is one of the tidyverse packages, specifically the tribble function, which this may be the only time we see it during the course, but it kind of facsimiles drawing a CSV file with individual columns. So you can kind of see there's a little squiggly with filter ID corresponding to that first column name, squiggly with dims corresponding to the second column name, squiggly with subset corresponding to the third column name. And then each of these next ones that's separated by a comma is each of the arguments corresponding to each of these column names, in which case we are creating a filter ID for singlets, the first gate, the second gate is going to be live, third gate is going to be T cells, fourth gate is going to be CD4, CD8 positive, double negatives, etc. And then you can see we're specifying under which parent each of these is being drawn, and the dimensions that are being used. So the same data that we saw above in each code chunk, but all together in a single view. What R is going to do is basically convert that into a Tibble, which is very similar to a data.frame style object, and it's going to actually be able to use those in interactively. So if you're going to use this approach to go ahead and load in all your gates in one interactive session, basically you just need to have a fresh gating set that's been transformed. So just rerun these lines of code. And then once that's done, we're going to use the gsapply gating strategy function, in which case we're passing the gating set, and we're providing that gating strategy Tibble, which we had saved as the gating table object. And as soon as you run this line of code, it's going to pop up asking you to draw the singlet gate. So you can select polygon, draw your singlet gate, hit done. But rather than returning you to the console window, it's just going to proceed immediately to the next one, asking you to draw out the live gate, in which case select rectangle, draw your live gate, select done, and it will immediately proceed to the next gate, allowing you to draw the T cell span gate. Vice versa, it'll ask you for each of the T cell populations. You can use polygon and draw the gate around each of those sequentially, providing all the information that we need. And then sure enough, if you plot it, you can see that rather than have to go back and forth, you just got everything done in a single go. Now, going back to one of the questions we had in the chat, as you may have noticed, flowGate the gates are being applied generally across the board uniformly to all specimens. And we are very much limited, at least in our current understanding, of how to be able to customize these for individual assessments. Consequently, we really need to be able to visualize each of these specimens so that if we're accidentally cutting into the wrong location for a particular specimen, we can go back, regate that particular gate just to make sure we're not missing out on important bits of our data when it comes to how we're gating in R. Now, there are many different ways to visualize plots. We encountered some of these back in week 6 when we looked at how to use ggCyto for visualization. The simplest way to view all the gates that are being applied on a per specimen basis is the autoplot function from ggCyto. And then just repeating this for every single specimen that's present in our gating set. So this is the easiest way to explain it today, but it's not the most time-efficient manner in the long term. So as we go further in the course, we're going to learn how to iterate through our specimens that are present in the gating set and just basically send all those plot outputs directly to PDF files, which will reduce the need to create individual lines of code to check every specimen. We'll also look at other visualization tools that will allow us to compare the individual gates across all specimens in the same view, which is the best way of being able to detect, for example, is your baseline different from your SEB when it comes to those cytokine gates, and do you need to make adjustments accordingly? So this is a work in progress as far as how do we go about visualizing them, but this is a good initial place to start, especially this week as we are learning how to gate. So for ggCyto, you're just using the autoplot function. In this case, you just need to specify which specimen in your gating set you want to visualize. You're using the double square brackets in this case and the index position within the gating set. And the formatting visualization is going to be pretty basic. It's not custom tailored to your particular plot. It's not always properly zoomed in. So I'm just adding the theme underscore black white layer just to make it a little bit more visually appealing for this particular class. And you can see all the gates that we drew for this particular one become visualized. So we can see we have a singlet style gate that's been drawn. We can see where the scatter gate was applied. We can see that the CD tree span goes between these two locations. This is one of those examples where autoplot didn't zoom in properly, which is where some of the better visualization tools we'll see in a couple weeks come in, just to be able to properly zoom in and visualize them all in one go. And you can kind of see how the cytokine gate was applied, at least in the context of the specimen. So if you're checking your gating set, you just need to then go and check all different 6 by switching out the position index in the line of code, just to make sure everything looks kind of proper for the gates that you have created. And this should work for today, and we'll show the more sophisticated manners of reducing the amount of effort and changes of the code that you have to do manually each time as we go. So in summary, for this first part of the today, we have seen that flowGate can be used to facilitate drawing gates in a general manner for your main cell populations of interest, which is what we saw in this first introduction. A couple things to keep in mind, namely the gates that we are applying generally across all specimens, and we have limited ability to customize these to individual specimens as of what we know right now. Consequently, being able to visualize each of these specimens to make sure the gate was correctly placed is very important, because that not only gives us confidence in our data, but anybody reviewing our gates that were drawn in R will want to be able to see that yes, these gates were drawn properly. We did actually get the cells that we thought we were getting. We're not getting a mixed splice of multiple populations because the gate was off. All in all, flowGate is a very nice R package when it comes to implementing supervised manual style gates. There are still some areas within the package that can be improved on, but the beauty is that the open source nature of this package, that if you have a good idea, have some of the coding know-how or the time to figure it out, many of these ideas can be implemented and then extend the package functionality to be able to do cool, new, useful things that it wasn't originally capable of doing. All right. We are going to go check out the openCyto package next. And openCyto is also in Bioconductor. It is one of the sister packages to both Flow Workspace as well as CytoML. It works a little bit differently than what we saw for flowGate. Using gating template.csv files, which we can then customize the individual gates that get created. We can specify the various algorithmic gating functions that are being used, and it can give us a substantial flexibility when it comes to creating and implementing automated gating schemes. So one of the advantages of this approach, at least in theory, is that when we provide proper constraints, it's better able to handle some of the interspecimen variability differences that we might encounter in our data due to batch effects that might otherwise have been thrown off by the fixed manual gates that's applied equally across specimens in the context of flowGate. However, there's something to remember that's one of the main truths when it comes to anything in computational cytometry. Namely, garbage in is equal to garbage out. So if you're just applying a default and you're not actually visualizing, you're not actually checking, you're going to get caught in a situation where you're not actually showing what you think you're showing, and the data is just going to be flat out wrong, and you're not actually leading to discovery of biological truth. And if there's anything else, always, always, always check these outputs visually, because I would hate to write a letter to your editor saying the R code was not implemented correctly. All right, so how does a openCyto package work in terms of this gating template? So I have included a gating template within the dataset that was provided for this week's course. It's already fully written out. It works properly for SFC files provided. So we first need to find this .csv file, and the way we can do this is we just need to switch out our pattern argument within the list.files. So we're still looking within storage location, but instead of looking for .fcs, we're now looking for .csv, keeping the full names argument set to true. We're assigning this to the object variable rGate template, which we're then using with the read.csv function to read in the CSV, just so we can visualize and see what's actually being shown. And this is what a openCyto gating template CSV is going to look like. Now, please note, if you didn't have your console window properly expanded, if it's something that has many different columns, it's going to repeat down subsequently down here. So we can see for this CSV, the column names are alias, pop, parent, dims, gating method, and then you can see there's individual rows 1 through 6, but then right below it repeats again, gating args, collapse data for gating, group by preprocessing method, all those being column names, and then repeating values corresponding to those individual columns for each of the rows being present below. So each gate that's going to be drawn by the automated gating method corresponds to an individual row that's present within these .csv files. So for our first example, just like we did for flowGate when we're doing the manual approach, let's go ahead and draw a singlet gate for our population of interest. We'll switch out and see how we can do clustering based on density and how we can do clustering based on separation between populations in a couple minutes. But for now, we're going to draw a singlet gate, which is going to look kind of like this. I apologize, I need to zoom in a little bit more for the slides. And what we're going to basically need to provide to our gating template is some of that same information we already provided when we're doing the flowGate, but this way we're providing it to individual columns. We need to provide a gate name, which is going to go to that alias column. We're going to need to specify to which parent gate does this belong. In this case, it's going to be the parent column. And then our particular x and y dimensions that we want to visualize will be going to the dims column. And then the gating method is basically where you're able to switch out which algorithm is doing what kind of gating. In this case, since we're going for a singlet plot, we're going to use the gating method of singlets that will allow us to draw a polygon around our particular population. So if we look at this first gate that we're creating, we can see alias is going to show up for singlets. Parent is going to show up as root. Pop kind of basically denotes, do you want the cells that land within your gate or outside your gate, which you would use in the context of an exclusion style one. In this case, pop is set to positive, so we just want the gate cells that are present within the gate. Our dimensions being used are forward scatter A by forward scatter H, notice the comma. And then the gating method, i.e. the algorithm being used, is the singlet gate specification here. And then these last several columns, gating args, collapse data, etc., these are defaults that are not used by every gating method, so you may not actually need to use them for your particular context. So we'll see when these changes occur as we go through the different gating scheme. And some of the same information we covered. All right, so if we look at our entire gating template CSV, we can see there's 6 rows corresponding to 6 gates that we want to automatically create, corresponding to singlets, to lymphocytes, to live, to T cells, CD4, and CD8. So let's go ahead and quickly check to see what a couple of those other gating algorithms are and how they're actually being applied. Remember, for all these different rows, the main things that will change on a per row basis are the name of your gate, the parent gate it belongs to, and your axis dimensions, leveraging your floor marker information to visualize different combinations that are then being used. So this is what it looks like currently. You can see we just drew the singlet gate. flowClus is a different gating method we'll use for lymphocyte population, and then the other gates are all mostly gate min density in terms of the algorithm choice. All right, so let's look at this flowClus gating method. As you can see, this looks pretty typical for what we saw for that first CSV row. The main difference at this point is we see flowClus is being shown under the gating method, and then there's some particular gating arguments. K is equal to 2, comma, and then target is equal to a little C for concatenate 2E6, so basically 2 million, and 5E5, basically 500,000 at that point. And then all its other default arguments remain equal. So this will generate a gate that looks something like this. And the way flowClus is working is basically a wrapper function to the flowClus package from R that's working in the background, which is able to determine where particular regions of density are occurring on your plot and gate around those regions that are separated from others. So the main thing when you're working with your own data is, you'll have lymphocytes here, you may have your monocytes, you may have your granulocytes, and then if you're particularly unfortunate, you may have a lot of dead cells and a lot of debris. So you actually want to be able to kind of point in the right direction of which of these little density regions do you actually want to gate. So that's where that gating arguments comes in. So we said that we provided a value of 2 million, which is kind of corresponding to this 2 million mark on the forward scatter. And then 500,000, which is kind of like right around there for the side scatter value. So you're basically saying, hey, look for a density region that's roughly around this point here, and go ahead and create a gate around that section. So that's what we end up seeing for this particular set of the gating arguments. Target is equal to C, 2E6, comma, 5E5. And then K is equal to 2 is kind of the internal for the argument goes. And this was mentioned in passing, is flowClus is using a density approach to figure out the clustering. There's several other algorithmic methods that are implemented within openCyto. I don't cover them all today, they're mentioned in the vignette, but this is kind of where you're able to bring in additional ones and change it based on what you're trying to do, what kind of parameters you're trying to work with. And this was mentioned of just make sure you're properly pointing at the correct location, because if it's doing density and there's no specification and you have more debris than anything else, it's going to gate around the density of the debris, not your lymphocytes. Right. And the very last type of algorithmic gates that you can apply with openCyto that we will cover at least today is gate min density. And you'll see that most of the gates specified within our CSV template are using gate min density as their method. So for live, for T cells, for CD4, and for CD8. What's changing for these is the individual dimensions, individual names, individual parents, but the gate min density looks pretty much the same across the board. So a good example of how a min density gate is applied in openCyto is looking at our CD4 population. So you can see it's pretty much following the defaults. All that's really changed is the name specifications for that new gate. But when it's actually applied, you'll see it generates a gate like this. So when it's drawing based on minimum density, it's basically seeing where your positive and your negative populations are, figuring out where the minimum density between these two is, and that's where it ends up drawing the gate. So obviously, this approach using an algorithm will work best for primary style markers, things that have both positive and negative cells present within the same sample. Obviously, if you're working with some of those secondary or tertiary markers, where it's just kind of like a little sliver over here and it never separates into a separate population, this won't be the algorithmic method that you want to use. You'll need to figure out a different option in this particular case. But this does a pretty good case example of drawing a separation between your positives and your negatives. Obviously, if you're drawing this by manual gating, you may want to tailor this over a little bit here, or vice versa, a little bit upper end. You can get the same effect by using the gate constraint arguments, which we'll see in just a couple minutes, but you can see it does a good enough job for this initial gate that's being applied. And in this case, basically, the pop, remember, was having it denoted as equal to plus. Basically, when you're working with a min density gate, a plus is equivalent to everything to the right side of that gate line. A negative is equal to everything on the left side of that particular gate line. So in this case, when we set CD4 and the pop argument equal to plus, we're basically saying keep the CD4 population that we find on this side of the line. All right, so what advantage do you have to using the gate min density as far as the gating method? It's very, very fast. So If you're using flowClust, you'll notice it takes a little while to like actually do the calculations of where the density is, gate around the appropriate level of your population, and then proceed to the next sample, repeating for each specimen. Gate min density is extremely fast, so as long as you have markers that are amenable to the gating method, you can get a lot of gates done in really small amounts of time. So, in the case, however, let's say you're looking back at this plot here, and let's say the min density gate that's applied is really off. So instead of being placed right here, it mistakes some little bits of debris or antibody aggregates way over here as the actual population, and it draws a gate like right around there. Or vice versa, it draws a gate somewhere negative 10 to the fourth, and everything is appearing positive. This is one of those situations that it's not going to work quite well just using defaults. You actually need to provide a gate constraint, basically saying, hey, I want this min density algorithm to run, but I want it to search within the region specified by these 2 points. So like between 10 to the third and 10 to the fifth, or the corresponding R transform values like 150, 450, depending on what the values are of the transformation. And the way this is applied is, once again, you just come into the particular line for this particular gate that's being applied, and you can see we're using gate min density, but then under gate underscore range, you're providing another little vector saying place the gate somewhere between 1 E4, comma 2 E4, which constrains it to placing based on minimal density of that particular region. So this is a broad overview of what a gating template resembles, but let's go ahead and load it in and see what happens when we apply it to our gating set. So openCyto recommends that when you read in the .csv file that you use the data.table package, specifically the fread function. The reason for this is it's a little bit better at enforcing formatting than compared to either the readr or the read.csv functions. So this is the one that you should go ahead and use when you're loading in your gating template. So what this looks like is we're just providing the file path to our .csv file, to data.table, to the fread function specifically. I will generally, only time I'm using data.table in my regular workflow is when I'm running fread, so I'm just going to use the double colon, basically not loading data.table, just going straight to the function. This allows us to then read this in as its own data.table. And once this is done, we can then pass it to the gating template function, which will wrap it correctly within all the different layers that openCyto needs to actually create the gates. So if we check to see what the output is by just printing the full template, we can see it's showing up as a gating template object with 7 populations defined. If we've been doing what we've been doing throughout the course and we just want to check behind the curtain, we can run SDR. We can see all those little @ and all these little $signs, which just tell us that, yeah, it's an openCyto style S4, S3 object. It's a little complicated. It's doing a lot of the work in the background for us. This is kind of analogous to what we saw in the console window readout when we were doing the flowgates themselves. At that point, if we check class, we can see that, yes, this is a gating template object from openCyto. And all we really need to do is have a fresh gating set that doesn't have any gates applied. So we're just rerunning all those lines of code that we saw before when we're setting up our gating set correctly. And then when it actually comes time to applying our openCyto template, we're using the gt_gating template. We're applying it here. And this is what a typical openCyto readout is going to show. It's going to start gating for that very first line, so gating for singlets. It applies all those gates to all the specimens, and then it gives you a little done readout sign. It will then proceed to the next gate, so you can see gating for lymphocytes. This is the one that takes a little bit longer in the calculations, so it takes a little bit. This is the printout for every single specimen, and then proceeds. And then this is when it hits those min density gates, and you see live is done, T cells is done, CD8 is done, CD4 is done, all is applied rapidly across the board. Now, if you have something that doesn't have any cells below it, it's going to return a little error, which is what happens here when there was no particular cell below. I think it was, maybe it was like below the CD4 gate looking at TNF alpha interfering gamma for one of the squadrons. And once again, it's always important when you're doing anything in R just to make sure you're visualizing the data, making sure everything that you think you're applying is actually being applied correctly. So let's go ahead for now, just use the ggcyto autoplot function just to get a quick visualization of what's going on. And once again, we're just having to check for now individually. And we can see that these gates have been applied. You can see our singlet gates. You can see the flow class didn't quite gate on all the cells that we might be interested in. This could be something we could adjust with the particular arguments being applied. But then you can see CD3, CD4, and CD8. It's a good enough approach, at least, to quickly delineate out the populations as far as positive and negative goes. And very similar to what we've done for flowGate, you can go ahead and save this gating set to your local environment so you can load it back in later using the save_gs function. Alternatively, because this is in a CSV template, you could just rerun this each and every time. So if you're using mostly min-density gates, it's going to be pretty faithfully replicable for what you're doing one time versus the other. The exception comes when you're using a lot of flow class algorithms for individual gates, because those are more stochastic, so there will be a little bit of variability of how the gate gets drawn. So this is something that you should experiment and see as far as like if you want to try to recreate it from scratch each time versus you want to do it once and then keep it there and save it and continue on. All right, so our takeaways during the course of this session, we have explored how to add both manual and automated gates using two bioconductor packages, flowGate and openCyto. With the examples that we showed today were introductory and they were separate since they both end up adding to gating set objects. We can also combine these together to do rather complicated gating schemes targeting cell populations of interest for our respective data sets. Separately, I would be very much remiss if I did not mention a couple additional packages that, while still under active development, are available right now via GitHub, and they may be useful to you in the long run. These are the Cyto Explorer R package from Dylan Hamill, as well as the Flow Magic from Sebastiano. We'll cover these on one-off sessions later in the course, but depending what you're trying to do with your own data sets, they may be worth checking out beforehand. Next week, we're covering the last session within the Introduction to R portion of the course, before we proceed into the more intermediate cytometry core portion. Namely, what we'll be covering next week is how to create your own functions, i.e. the tools in our tool chests, if we're following the analogy we've been using throughout the course. This was an area that when I first was getting started, I didn't actually realize quite how important a skill set it would prove to be in the long run. So to better set you all up for success for your own adventures, I want to introduce them now so that you can figure out how to use them. What we're going to be trying to doing is basically set up our own functions, our own tools, that will allow us to derive the cell concentration from our acquired FCS files based on a particular gate. And that's all I have for now. We're closing at the top of the hour, so if anyone needs to take off, feel free to do so. In terms of additional resources, both of these packages are from Bioconductor, which means they have really good vignettes. And when it comes to how to use these packages to their full extent, there's no one better to the explanation than the maintainers themselves. So I'm referencing back to the flowGate introductory vignette. We covered most of it, but there was a couple points we didn't have time to touch on. So you may still circle back and double check on these. openCyto, there's multiple vignettes for openCyto. The first one's more introductory. We covered a good portion of it, but there's still additional versions of gating methods that we didn't have time to cover for more of those secondary, tertiary style markers, so it may be worthwhile checking out on your own end. The third and fourth vignettes, respectively, are how to use different auto-gating functions, because there are many different gating methods. So the question was asked about Boolean gates. There's a mention of how to implement these within the context of a .csv template within, I think it was the second vignette here. And then how to write a CSV gating template. Some of these more complicated combinations can be a little weird on the syntax as far as putting them into each respective line of a template. So this is a good place to go for reference for this particular aspect. Our take-home problems for today is, especially if you're planning to switch over to R long-term for your gating, a lot of these skill sets that we covered today are going to be essential, especially if you want to be confident in your own abilities to adjust the gates, figure out when something's not working, and implement the fixes that you need to do. So the first take-home problem for this week is basically using flowGate, using your own data set or R data set, whichever you want to do. Just go crazy, create as many gates as you want for all the populations that you may be interested in. Go ahead and save them to your local environment, and then load them back in. Add another gate, intentionally mess it up, figure out how to delete it, just get very comfortable with how to use flowGate. Same thing for openCyto. These gating templates take some while to get used to as far as writing them out for new gates, but once you do, they can be quite fun to do because they can speed up things and make it much easier on your end. So go ahead and play around with setting up an openCyto template and making sure you're able to reload it again. And then the third one is we've kept flowGate and openCyto kind of separate, but just go ahead and take the openCyto template that you generate, and then on top, save a few additional flowGates and save it out just to make sure you can kind of combine the best of both worlds when it comes to both manual and automated gating, fusing them together, allowing you to build much more complicated workspaces in general. And that is all I have for you guys today. I will see everyone back next week when it comes to how to build our own functions. The compensation one-off, which was still left to do for those who are doing conventional flow cytometry, will be coming out in probably the next couple days. I'm wrapping up a last little section of that discussion. The community feedback survey is still out. Everybody should have gotten it in their inboxes. I'll keep it open through the end of this week. And with that, that's all I have for you today. If there's any questions, I'll be in the chat for a little bit, and if not, I'll see you on the discussion forum.